Chromosome and cellular reproduction. Today, I'm going to talk about prokaryotic cells, eukaryotic cells, what their chromosomes look like, and what about viruses, how they do reproduce, and how their functions differ from each other. What I mean by functions is how they behave, okay, how their genome is structured related with each other. Those kinds of things we're gonna focus in couple of future lectures. But in this particular lecture, what I'm going to discuss I'm going to discuss about chromosome and the reproduction of prokaryotes, eukaryotes, and how viruses reproduce. If you need more information, you can go to the Pierce Genetics 5th edition to get more information regarding chromosome and cellular reproduction. Let's start with the general concept about prokaryotic cells and the eukaryotic cells and virus cells. In my previous lecture, I discussed about the difference between prokaryotic and eukaryotic cells, and but I did not mention about any virus cells in my previous lecture. But today, I'm gonna just like shortly review the different structures that we can see in prokaryotic cells and the eukaryotic cells with the virus cells. Let's start. The prokaryotic cells are unicellular, but eukaryotic cells may be unicellular or maybe multicellular. Okay, and the virus cells are acellular, as you know that. And if you know the eukaryotic cells, you have membrane-bound cell organelles, but in prokaryotic cells, you don't have anything. But if you see the virus cells, you don't have any kind of inner contents. Look, nothing. You just have DNA, okay? But you cannot have any chloroplast, any kind of other cellular organelles like mitochondria. You cannot see any enough structure in prokaryotes and like in bacteria in like prokaryotes, and in the case of the viruses you cannot see any inner kind of content here, right? So basically it is very difficult, okay, how to understand the complex mechanism how they do go on the metabolic activities to sustain their life. That's very complex. They do not have enough kind of like this kind of structure, okay? So I don't know, we're gonna focus on that, don't worry about that, okay? Let's see. They do have ribosomes, eukaryotes and prokaryotes, but their units are different. For example, in eukaryotes you're going to have 80s ribosomes, but in the case of bacteria or in prokaryotes they do have 70s. 70 of ribosomes are made up of, as I told you, the 50s and 30s, okay? And for the eukaryotes, the ADS is made up of 60s and 40s subunits. Those subunits are composed of rRNA, okay, and those are composed of some kind of proteins also, okay. So that's important. And one main interesting fact about these kinds of cells is to understand by looking their mechanisms and how they correlate with each other. As you can see here, flagellum, which is a locomotory organ, which is more common in bacteria, also in eukaryotes also, okay. And these flagellas, not only flagella, they may have cilia also, okay. These are like locomotory organs, okay. So but the viruses here, if you look, these are very interesting creature, okay, and these are very important, but they do cause some different kind of diseases, but these are very important. This outer membrane is the capsid, okay. The inner is nucleic acid, like which may be like DNA or RNA, any kind, okay. To survive, not to survive, to reproduce, they need host. they can survive in inert place like without host for like thousands of days, so that's why very interesting enough. As I told you, the prokaryotes are very smaller in size, which are 1 to 10 micrometer in diameter, while eukaryotes are 10 to 100 micrometer in diameter. Okay, and the eukaryotes possess usually linear DNA, which are complex with histone proteins, but they do not have, the prokaryotes do not have histone protein except some archaea. They do possess histone, but their chromatin structure is quite different from the eukaryotes, though. Okay, so those are the basic difference we just covered. Let's look some table here. Look, okay, this will explain very well. These are the things I just mentioned. Okay, uh, I didn't mention the cytoskeleton. In the case of prokaryotes, it is absent, but in the eukaryote, it is present. Okay, and this is a quite very like a small review, like kind of summary. The 70s ribosome in prokaryotes, okay, which is larger is 50s, as you see here, right, and smaller is 30s subunits, and 80s, which is in eukaryotes, which is like 60s and 40s. But remember, if you just combine 60 and 40, it's gonna turn out to 100, right? So it doesn't work well like that way. Okay, so sometimes it is not a addition rule. Okay, you need to think of that. As I told you, these rRNA and the proteins are very important in subunits, like smaller subunits. Okay, these are like in present in different proportions. Sometimes proteins is higher, sometimes rRNA is higher. Okay, they just like work together. Okay, eubacteria does not possess any kind of histone protein. The histone proteins is very important. Histone protein is very important, which I'm going to cover in my next future lecture. And but archaea, they do possess histone protein, as I told you. All eukaryotic cells are complexed with histone proteins. Means histone protein, there are different types of histone protein, H1, H2A, those kind of things. Okay, we're gonna discuss in future lecture. Uh, there are some questions. Think before you move on. What do you think about the circular DNA? Why circular DNA is very important? Okay, the main reason I think the circular DNA is very important because it is more efficient. Okay, circular DNA is more efficient. Okay, what I mean by more efficient is there is no telomere. Okay, no telomere means there is no require of energy. Okay, that is energy is very important to keep stabilize the telomeres end. The telomeres in like keep like losing every time because those are the ends. So to protect those ends we need energy. So if that is circular DNA, you don't need to protect those ends, so no loose ends. Okay, so that's why I think it's very important. But in the case of like multicellular organisms, circular DNA is not like more common because sometimes it is more problematic. I'm so sorry, problematic. Okay, so maybe there may be very difficult in the case of replication. Okay, so that's why we do not see usually circular DNA in the case of eukaryotes, but due to some mutations may occur. So the mutations may change the DNA structure. Okay, so just I discuss about that, why histone proteins are important. So just keep in mind, just now is just histone proteins are very important in forming nucleosomes. Nucleosomes is basically is a octamer, is a forming of octamer in which the histone proteins are binding, it's clamping the DNA strand uh, like as a clamp. Okay, so that's why histone proteins are very important, and it is basically related with the gene expression. Okay, how tightly it is packed, what kind of nucleosome, like what I mean by what kind is like uh, how far is nucleosome, what is the position between nucleosome, okay. Those kind of things are related with gene expression, which we will discuss in future lecture. Let's talk about some reproduction in prokaryotes and eukaryotes. The prokaryotic reproduction is basically through asexual, but they may go through sexual reproduction also. Okay, it is not all the time asexual. They may go through sexual also. Uh, in the case of, let's say, in prokaryotes, the binary fission is very important, in which like the cells grows and the replication takes place. How the replication takes place, we're gonna talk about in just few moments. Okay, so the cell will grow and the cell will divide in equal half, and which is called binary fission, and it is very important. But in the case of binary fission, you're not going to see any kind of genetic diversity. Okay, no genetic diversity you're going to see there. So no genetic diversity. Okay, so no genetic diversity through binary fission. There may be transformation or transduction. Transformation means like just like transfer of DNA is called transformation. Okay, and the transduction is by the help of like kind of viruses. Okay, when the genes are transferred, then that is called transduction. And the conjugation is basically is a type of like sexual reproduction. Okay, in which DNA is transferred between prokaryotes, okay, by means of pilus. The pilus, which is pili, which is a hair-like structure, which pronounces P-I, okay, L-L, okay, I, pili, or you can say pilus, okay, whatever. So it is a hair-like structure, appendage, which is found on the surface of the many bacteria. The pili joins with each other, and they do transfer of the information. So it is the only one way they do sexual reproduction. But in the case of eukaryotes, okay, so two types of reproduction for eukaryotes, which may be sexual or asexual reproduction. Okay, you're gonna see some kind of mitotic reproduction and meiotic reproduction. For example, if we have like kind of, let's say, 2n, okay, and the parent is 2n, and if you get the children copies which are like in the same amount in a diploid, which is also called diploid, 2n 2n, then that is called mitotic. Okay, and the meiotic or meiosis means you're gonna get like diploid parent. Okay, but you're gonna get the haploid. Okay, you're gonna get the haploid offspring. Then that is called what? That is called uh, meiosis or meiotic cell division. Okay, and the meiotic cell division produces non-identical gametes. What I mean by non-identical means they poses like crossing over, like changing interchanging of the genetic information, which may cause, there are like different kind of things may happen, like non-disjunction, crossing over, like formation of chiasma, like different kind of things may happen. Mutation may happen, which may alter the whole information. Okay, information may be like carried out in wrong way. Okay, so there is a variation you're gonna see here, but not and a variation in the mitotic kind of reproduction, which we will cover in the future in brief. Okay, let's talk about the very important role, um, how prokaryotes do divide. Okay, so how they form offspring by means of binary fission, let's say. In the case of prokaryotes, the reproduction starts, okay, very important enough, the reproduction like of prokaryotic cell, which contains a single circular chromosome or DNA. What will happen here is they do possess the origin of replication. Origin of replication means from where the replication starts. Okay, so basically replication, it starts at origin of replication. Okay, and what will happen is the cell will grow. Okay, there will be like two replication origin. Okay, one on each side. Okay, and what will happen is you're gonna see the cleavage like a structure from the middle, and you're gonna form like two identical new copies. Okay, so DNA replicated into two. Okay, and each origin is like separated through the membrane, like forming of cleavage and those kind of things you're gonna see. Okay, so this is about the prokaryotes. And if you look about in the case of eukaryotes, it is very interesting. The chromosome structure is interesting, and different kind of like organisms possess different number of chromosomes. For a human being, 46, right? So we know that. Okay, and remember one thing, like each gene possesses two allele in most of the cases. Okay, for example, like hair color. In hair color, like two gene, okay, are responsible for hair color. Okay, so that is important. Before we move on to there, how chromosomes are formed, this is a basic DNA structure. If you see here, this is DNA. Okay, so DNA, double helix DNA, which is antiparallel, and you're gonna see nitrogen bases here. Okay, what will happen is the DNA gets more condensed and condensed and condensed, and finally it poses like too many, like very thick kind of structure, which we call uh, the chromosome. Let's start from here. Okay, so let's start from here. At the simplest level, the chromatin is a double stranded helical structure of DNA, as I told you. Simple. Okay, Watson and Crick proposed this one, right? So you're gonna see like double stranded here. What will happen here is it will like further condensed, and there you go. You're gonna have histone. Look, histone H1, as I told you. Histone clamps this strand and forms the structure called nucleosome. Okay, and what will happen is this nucleosome further condensed and form 30 nanometer fiber. This 30 nanometer fiber that becomes loops overlapping like 300 nanometer, you're gonna see here. Then it further condensed, you're gonna see like for the 700 nanometer and so on. This process is very complicated. Okay, coiling, and you can have condensing DNA structure by forming nucleosomes. Not only nucleosomes, you're gonna form more condensed structure, and you're gonna form 1400 nanometer type of like kind of structure called chromosome, and it is very important for the reproduction, either mitotic or meiotic. Okay, so this chromosome, which may be metacentric, which may be submetacentric, acrocentric, or telocentric, how we gonna recognize what kind of what type, okay, of chromosome it is? Okay, so we need to concern about the centromere. You see that this dot here, this is the centromere. Okay, this centromere poses kinetochore, okay, at which spindle fibers attached. We're gonna briefly discuss in the mitotic and meiosis kind of cell division later on. Okay, spindle fiber, their structure, those kinds of things. Metacentric means you're going to have two arms. As you know, the chromosomes are labeled as P and Q. One arm is P and another arm is Q. The P arm and the Q arm, if they are equal in size and the centromere is at the middle, then that is called metacentric. Okay, if two, like one of the arm is longer than the another one, then that is called submetacentric. And the acrocentric is like not on the tip of the telomere. Okay, this is telomere, but just leave like little space here. Okay, so this is the space that is like left over little bit, and that is the acrocentric, almost at the end but not at the end. Okay, telocentric means there is no one arm. What I mean by that is you're not gonna see P arm here. You have only Q arm. So only centromere on the tip is called telocentric, and they're like very important enough. Okay, it may be due to some kind of maybe inversion or those kind of thing like loses of that chromosome structure. Okay, if you lose some part of the chromosome, that means you deleted a lot. Okay, so that we're gonna discuss in future lecture also. Okay, let's talk about very important why. Okay, what is required to become functional chromosome? Uh, why centromere is very important? Why telomere important? Why origin of replication important? Let's see that. Okay, what is required to become a functional chromosome? First is centromere. Okay, if there is no centromere, chromosomes will not move. Okay, so moving chromosomes during the cell division is facilitated by centromere. Okay, and telomeres are very important because they protect and stabilize the chromosomes like ends, the origin of replication where DNA synthesis begins. As I told you, as I discussed before, you can have cell structure, you can have origin of replication on the both side. The cell will shrink, it's gonna form like cleavage like a structure like this way, okay, and it will divide. Okay, in the case of prokaryote, right? You're gonna have one half here, one half here, right? So you're gonna have identical copies here. Okay, so why it is very important? The kinetic core in the case of centromere, as I told you, okay, which is a kind of protein, okay, what will happen is this spindle fiber, which consists of microtubule, it attaches with the inner plate, okay, and when it attaches to the inner plate, it will help for the detachment of like chromosomes. Sometimes they have to move during anaphase, they have to move during metaphase, they have to come together on the metaphase plate during cell division, right? We're gonna talk about those briefly. The another very important concept we need to think of is cell cycle. Okay, cell cycle is very important and it is very complex process, and it requires a huge knowledge to understand the concept. The cell cycle and the life cycle is different though. Okay, cell cycle means how cells goes through inner phase, okay, to mitotic phase, okay, how it goes. Okay, so it is very critical process. Okay, let's talk about that. Let's talk about first inner phase. What is inner phase? Inner phase is a first stage of cell division. Okay, so inner phase, as you can see, inner phase is this part is inner phase here. Up to like this part is inner phase. Okay, the inner phase part consists of G1, okay, which is here. Okay, it consists of S phase. It consists of G2 phase. Okay, then you're gonna have M phase. Okay, M phase you're gonna have mitosis. Okay, after mitosis, this mitosis also includes prophase, prometaphase, metaphase, anaphase, telophase, and eventually cytokinesis. Okay, then you're gonna have another cell. Okay, so this process will happen. But remember, as I discussed, like as I say, like G1, S, G2, it is not like it's easy like that. You're gonna have different checkpoints. You're gonna talk about checkpoints. Okay, those checkpoints are very important also. We're gonna briefly discuss those. Okay, let's talk about first G1 phase. G1 phase is very important. Okay, it is very important. What will happen at the G1 is cell grows. Okay, cell grows very efficiently, it grows. Okay, and it will pass through the G1S checkpoint. We're going to talk about different checkpoints, okay, uh, in few moments. The G1 when it enters to the G2, so S phase, sorry, it must go through G1S checkpoint. Okay, sometimes G1 may enter to the G0 phase, which is also called the stabilized phase or it is non-dividing phase. Sometimes when cell goes on this is like stage, they do not come back. Okay, up to like until you die, they're like in the same place. Okay, they do not come back to the G1. Sometimes they do come, like for example, neurons. They do not divide, right? Non-dividing phase is called G naught. When everything is fine at G1 S checkpoint, then the time is S phase. Okay, as I told you, G1 S checkpoint, the cell is committed to divide. If everything is good, cell is well enough grows, okay, for the division, and everything necessary for cell division is there, then you may not have any reasons to stop there. You're just gonna go ahead, okay, complete the process. In S phase, the DNA duplicates. Remember, as I told you here, DNA duplicates, but chromosome do not duplicate. That's the concept. Here you go. Sometime in exam you may have like, okay, let's start. A species has 4 chromosomes. Okay, how many chromosomes in S G2 phase? You may think it is double. It is, I say chromosome, not the DNA. So chromosome number will remain same into G2. But if I asked DNA, 4 DNA molecules, then you're gonna have here 8. Okay, because you're gonna double that. Okay, basically in S phase the duplication will take place, and the after duplication the G2 phase you're gonna have different protein synthesis, biochemical reactions takes place, you know, like different kind of things happen, and whatever cells need everything will be prepared, and the G2M checkpoint makes sure that everything is very important. If something is missing, the cell gonna die here. It's not gonna proceed to the further. Okay, so it will stop here. It will very important. It will play a very important role. Okay, then it will enter to the like mitosis, which is a M phase. Okay, the mitosis, as I told you, there is prophase, like metaphase, anaphase, telophase, and after that the cytoplasm divide, cytokinesis takes place. Then those kind of thing will happen. After cytokinesis, another new cell will produce. That new cell again go through G1, go through S, G2, and again this process will repeat whole time. Okay, this is a general concept we just discussed. Let's talk a little bit brief. As I told you here, the G naught phase is a stable phase and it is non-dividing phase. Okay, the inner phase has 3 steps: G1, S, and G2. G1 growth and development takes place as I told you, and you're gonna have G1 S checkpoint, and the S phase will be there, where synthesis of DNA duplication takes place, right? In G2 phase, preparation for division. Okay, you must have different kind of enzymes, proteins. It is not easy. Okay, so it is a very important role for the G2 phase to carry out. Okay, G2M checkpoint makes sure everything is good, then the cell committed to go through M phase. As I told you, prophase. Don't try to learn this. Okay, I'm going to talk briefly about M phase in my future lecture. Okay, although the length of inner phase varies, okay, G1S and G2 phase, cell like takes longer time in one phase than another. Okay, usually a like typically dividing mammalian cell spends about 10 hours, okay, in G1, okay, 9 hours in S phase, and 4 hours in G2. Why on G1 it takes longer time? Maybe the cell growth takes growing, is like very important here. Okay, cell takes longer time to grow. Okay, how many years do it take to grow? It depends like that way. The cells growth is like very important. Okay, it goes one by one. Okay, so it is very important. It takes, that's why I think it takes longer time. Okay, but the reasons are like still not explained well. Okay, in the case of checkpoints, there are different checkpoints. Okay, G1S checkpoint. Okay, you're gonna have, okay, G1S checkpoint, G2M checkpoint. Okay, so these checkpoints basically make sure everything is very good in proportion, so the cell can go without any confusion, any kind of problem to the next step. That is the main step, main thing we need to think of. Okay, okay, so G1 phase is here, S phase is here, G2M, cytokinesis, and again G1 phase. Okay, you're gonna have some CDK, it is called cyclin dependent kinase. Okay, and you may have cyclins also. Okay, cyclin and cyclin dependent kinase. Okay, in my next slide you're gonna see that. Okay, now this slide is very interesting and very organized. Okay, if you see here, this is a cell cycle we just discussed. Now we basically focusing on the checkpoints. Okay, if you look here, you're gonna have cyclin dependent kinase 1 at M phase, right? But we're gonna start from the G1 phase though, right? In the G1 phase, look here, you have cyclin dependent kinase 4 or cyclin dependent kinase 6. Okay, and after that you have cyclin dependent kinase 2. Okay, you can have cyclin E, cyclin A. You can have like cyclin is like, you have different kind of cyclin too. C, D, E, A, B, H. Different kind of cyclins you have. Okay. Also you have cyclin dependent kinase, different kind of cyclin dependent kinase, and they pair with each other. If you look here, this green and the red are like pairing with each other, because without cyclin dependent kinase cannot be functional. Okay, cannot be activated. So why it is like that? The cyclins needs to combine, okay, with CDK. What kind of, I don't care what kind of CDK that, okay, to form a functional unit. What will happen is cyclin dependent kinase are the enzymes. Okay, this depends upon the cyclins. That's why name comes from there, you know. So when they pair with cyclin, they get activated. Okay, when they get activated, they, what will happen here, look in the next picture, when they gets activated, the phosphate will activate it, and it will bind a phosphate. Okay, then what will happen, then this cyclin dependent kinase which is activated now will phosphorylate other. Okay, remember, cyclins, cyclin concentration and the CDK concentration throughout the cell is different. What I mean by that is it is not always same. Okay, CDK is, CDK concentration is always same in the case of cell division, but cyclin concentration is always changing. Okay, and there are different kind of cyclins as I told you, and as I told you, CDK depends on cyclin, and it gets activated. The activate, and it activates, which the CDK which is activated, which activates other by phosphorylating them. Okay, so that is very important. And different cyclins and different levels of phases of cell cycle. You know, Like different cyclins are playing different roles in different phases. In G1 there is different cyclins, G2 different cyclin, S different cyclin, right? You can have different types of cyclin there. Okay, that's important. And if you see here, okay, there are V1. This is the inhibitory kind of kinase. Okay, it will phosphorylate, but there is dual phosphorylation takes place. Okay, uh, which I'm gonna cover in few moments. Okay, why dual phosphorylation? Why V1 is inhibitory phosphate here? We're gonna talk about that one in few moments. Okay, let's go on the another slide. Okay, okay, let's see. If you see here, cyclin concentration is varying, and as I told you, CDK is always constant. Okay, when the cyclin in G1 cannot work for S phase. What I mean by that is cyclins gets degraded in each level and new cyclins are produced. New cyclins are formed. Okay, and as I told you here, the peak value here, cyclin E, like on a G1 phase, it is like higher and it goes down. Another cyclin goes higher, again down. These kind of things are going on here. Some kind cyclin dependent kinase, sometime off, sometime on, sometime off because there is no cyclin. Without cyclin, cyclin dependent kinase cannot phosphorylate other kinase, so it gets like turned off. Okay, so you need cyclin, different kind of cyclins to proceed. Okay, let's look this. This is cyclin, and you have cyclin dependent kinase here. Okay, the cyclin basically binds, okay, binds to the cyclin dependent kinase. Okay, and these two will form a similar small, like kind of like complex structure. Okay, when this, okay, now it is activated, CDK is activated, right? Now it it gets phosphorylated, right? What will happen is there is a dual phosphorylation will take place, as I mentioned before, right? Dual phosphorylation. What will happen is there are two kind of kinase which is called CAC. Okay, CAC and V1. CAC and V1. What is CAC? CDK activating kinases are called CACs. Okay, and these CACs phosphorylate CDK. Okay, these CACs phosphorylate what? CDK. As well as V1 also phosphorylates CDK. Okay, remember here. Now, when it gets, you have a cyclin here, okay, you phosphorylate through CAK and V1, which is called dual phosphorylation. Remember, until V1 phosphorylating is present here, this CDK is not active. You need to remove phosphate. Okay, you need to remove phosphate. Okay, and which is done by CDC25. Okay, which removes phosphate and turns inactive CDK to the active form. So CDC25 inactive form go and bind to the site. It just cleaves the phosphate. Okay, this inactivating the phosphate. Okay, the inactivating phosphate, uh, which is just like it. it just takes over, and the CDC25 just bind with that, and the phosphate will remove. And the CDC25 activate is a phosphatase enzyme, okay, and it is very important. And in the note, cyclins and CDKs must work together, which triggers next phase of cell cycle. Until after after the activation, okay, it will phosphorylate other, it will produce some kind of sagostine and those kinds of like enzymes or proteins, right? So which may be essential for the separation of chromosome, chromatids, those kind of things, right? In future in cell division. In addition to that, here, if you see here, you're gonna have positive kind of uh, control feedback here. Positive feedback. Why positive feedback? What I mean by that is, for example, okay, CDK binds to cyclin, right? You're gonna form an active form, but sometime what will happen here is CDK will bind to cyclin, okay, and you have V1 here. The V1 here will not activate this thing. It cannot phosphorylate other the CDK. So what will happen is CDC play a very great role. It cleaves that phosphate and it will make this functional. Okay, so this is positive feedback. Okay, if you see here on the time scale here, and the protein concentration is here. Okay, it is a CDK protein. Okay, so what will happen is cyclin protein is like this. The CDK activity is like going down on the time. Look, CDK, when it will go up, okay, it will go up, and eventually CDK activity is going up, okay, because the CDC is like helping that, okay. So that's why it is very important. We need to think of that. Remember, turning off cyclin CDK. CDK and the cyclin all the time may pair with each other, which may phosphorylate all the time, but you don't need all the time phosphorylation, right? Sometimes you need to stop the cell division, okay, which is done by two processes, which is called ubiquitin and proteasome. These are the two things, okay, from which, okay, the cyclin CDK will turned off, and there is no more further phosphorylation, no more cell division will occur. Okay, if something wrong, maybe ubiquitin and proteasome will stop the cell division. Ubiquitin is a kind of protein or which just like come and just bind to the cyclin, and it will not allow or it will just detach that, and it will just like form a regular kind of thing. It will degrade it. Proteasome, look, degradation will take place. You're going to see that kind of thing. And sometimes there are like mitogens. The mitogens are proteins, okay, or peptides that induces cells to start the cell cycle. The mitogens start the cell cycle, but anti-mitogen stops the cell cycle. Okay, so these are the just opposite. So mitogens bind receptors that trigger a signal transduction pathway that leads to the activation of cyclin D. As you can see, the mitogen, okay, which triggers, okay, a signal transduction pathway that leads to the activation of D, okay, cyclin dependent kinase 4, okay, and the cyclin dependent kinase 4 then triggers the activation of E cyclin dependent kinase 2, which you can see here. Then eventually the final one, cyclin dependent kinase 2 and the cyclin A gonna paired up, which eventually leads into the DNA replication. This is the kind of mechanism goes through it, and these checkpoints are very essential to uh, start or to make sure everything is right during the cell division. We discussed different types of uh, cyclin dependent kinases, and we discuss the mechanisms in detail. And in the future lecture, we're going to talk about mitosis and the meiosis. We're going to talk about the chromosome number and the DNA number in each and every steps during the cell division. And we're going to talk about the importance of mitosis and the importance of meiosis and the importance of meiosis in the genetic variation, and what is mutation, what may result in mutation, what is non-disjunction, and what causes non-disjunction. Those kind of things we will discuss. And basically we need to focus on uh, what kind of enzymes and what kind of proteins are very important. And look, the mpf maturation promoting factor, it is very important during the m phase. This is very important. Okay, this is the functional, it has the capacity to phosphorylate other or to form other kind of enzymes. Okay, so this kind of things. So that's it. See you on my next lecture. See you soon. Bye bye.